Hello everyone, my name is Ralison Manankasina, also known as Alex, and today I'll be presenting my research project titled Plastic Waste, the Full Power of PETase. I'll be specifically focusing on the expression, purification, and functional validation of PETase in E. coli K12. This experiment was carried at Holland College in Charlottetown, PEI, Canada, in the Bioscience Technology program. So why? Some context. Plastic pollution is one of the most pressing environmental challenges we face today. Polyethylene terephthalate, also known as PET, is basically the main plastic you can see everywhere, in the water bottles, in food packaging, but also in clothing fibers. And it accumulates in our ecosystems for hundreds of years. Why? Because, well, nature cannot break down plastic. Can it though? In 2016, a groundbreaking study by Yoshida and colleagues discovered a bacterium called Ideonella sakaiensis that naturally produces an enzyme capable of breaking down PET plastic. That enzyme is called PETase. But producing PETase efficiently outside of its native organism is still very difficult today, and that's basically where this research comes in. I wanted to express, purify, and confirm the activity of PETase using E. coli K12, also known as poop bacteria, as my production host, and investigate whether I could achieve production of PETase with lower concentration of IPTG than the standard that everyone is using. Basically, you have the E. coli, you put the plasmid DNA responsible of PETase production inside of it, but for it to be able to produce PETase, you need to induce it using a chemical known as IPTG. And I wanted to test if 0.5 millimolar of IPTG is as efficient as 0.1 millimolar of IPTG. So that leads us to my two hypotheses, which is first that the experiment would work, so that PETase could be successfully expressed and purified from E. coli K12, but also that 0.1 millimolar would be as efficient as 0.5 millimolar in IPTG for inducing production of PETase in E. coli K12. How did I do it? So we started by transforming E. coli K12 with PET21B, which is basically the plasmid DNA responsible of PETase gene. And then we induced protein expression with both two treatment level, 0.1 millimolar and 0.5 millimolar of IPTG. Before that, we confirmed that the plasmid was present in our culture by growing the culture on LB, agar, and ampicillin, because basically the plasmid causes antibiotic resistance. The transformation was done by electroporation. Yep. Once our bacteria was induced, once the production of PETase was induced by IPTG, we then added chicken lysozyme from chicken egg white and sonicated the cells to basically pop up the cells, centrifuge, and the supernatant of that is basically our PETase, technically, but we still need to purify it, and for that we used histochromatography using a nickel charged resin. And after all that, we collected all of our fraction and it was analyzed on STS page to confirm that yes, we did have PETase, and it's basically here. Those two fractions are my elution number 2 fractions on both treatment group, and these are basically all the debris and all the impurities that were present in the bind wash elute, in the bind wash fractions, and also in the pre-LPLC fractions. So how did we confirm that our PETase is working? Well, we used an enzyme activity assay. Here it was a kinetic assay known as PNPA colorimetric kinetic assay. So what's the result of that one? So this Two-way ANOVA was basically made to compare both treatment group, but also to see if the activity of the PETase that we got was statistically significant, or it was just due to, well, chance or random. And we could see that the two treatment groups were only significantly statistically different in the elution number 1 and in the waste fractions, but across all of the other fractions, they were basically the same. And the only fractions that were significantly different to the blank are elution number 1, elution number 2 in both treatment level, and the waste in 0.1 millimolar. But that is basically due to probably some PETase getting lost in the waste. So yeah. So what's the conclusion? Basically, both hypotheses were supported. PETase was successfully expressed and purified from E. coli K12, and also 0.1 millimolar IPTG is as efficient as 0.5 millimolar IPTG according to my studies. And this is significant because it basically suggests that we can reduce the inducer concentration without sacrificing enzyme yield. What will be the future directions of this research? Well, I didn't have time to do a plastic degradation, and that's basically the whole question here. So some plastic degradation using our PETase will be the next step, and analyzing quantifying the degradation by HPLC using MHET and TPA as standards will be a logical continuity to this research. And also, since we saw that 0.1 millimolar IPTG is as efficient as 0.5 millimolar, why not try lower concentrations to see basically the limit of what we can use, because IPTG is basically a bit toxic to the cells. So yeah. Well, finally, I'd like to thank Dr. Mike Gibson, Marla McKenzie, and Crystal McFadden for their invaluable guidance throughout this project, and special thanks to all my colleagues, especially Mr. Suleiman for his support. And yeah, thank you everyone. Don't forget to subscribe, and see you.